The purpose of this video is to give you a brief introduction to the scientific method. I realize this is something that you've probably heard to start off every science class ever since you were in like the 5th or 6th grade, and that everybody does it a little bit differently. But I'm going to go through and hopefully show you something that aligns very closely to what you learned last year as a 9th grader. But as far as the scientific method goes, the easiest way to describe this is that it's an organized strategy for problem solving. And this is something I'm going to try to show you in class. We'll go through and solve some simple problems, and I'll show you that problem solving strategies that you probably already use are things that fit into the scientific method. You just have to look at them in a different way in order to see it. To talk about the first step, though, of the scientific method, we'll be looking at some kind of a purpose or a problem. This is going to vary from topic to topic. Sometimes we'll just be solving a problem when it comes to your lab that you'll be completing. Other times there's a purpose that we're fulfilling for the lab. To give you an example of one of each of these, when we do the DNA extraction, once we get to genetics, for that lab we simply have a purpose. Our purpose is to extract DNA first from some bananas and then eventually from some of your cheek cells. We know there's DNA in there, so it's not like there's a problem. We're not trying to figure out, hey, do my cells have DNA? We know that they do. We're just trying to extract it. Other times there is a specific problem that you're trying to answer. Like the first lab that we'll do in class has to do with the properties of water, and your problem there is to actually go through and use the steps in the lab to prove various properties of water that we'll be talking about. So there's always some kind of a question, something that should get you thinking in order to drive the beginning portion of the scientific method. The next thing is to do some research. Now, you don't have to end up like this guy and be all stressed over the research. This sounds like a complicated thing, but I'm typically going to give you this. I'm going to give you some kind of background knowledge in order for you to base your hypothesis on. This becomes very important when it comes to the way that we're going to develop a hypothesis now that you are moving up in a few years at the high school. So things are a little bit more complex now. I'll talk to you about the way maybe hypotheses were done before when you're at the middle school and the way that's going to change a little bit for us now. But the important thing is the research is really what allows us to go through and change that. So since you're doing research, your hypothesis is going to be a little bit different. If I asked you to take a second and define a hypothesis for me, I would think most of you would come up with this answer. A hypothesis is an educated guess. And that's something that we definitely talked about in the past. Like I said, you probably picked that one up at some point at the middle school, but we're going to try to get rid of that idea. We're not thinking of it as just guessing because, as we talked about the step before, you're doing research. So you're going to base this on information. It's not just going to be a guess anymore. So the way we're going to go through and describe a hypothesis is we're going to call it something else. We're going to call it an informed prediction. It's informed because you're doing this based on information that you've already gotten from me in your research. Now, research isn't going to be something that's extremely thorough. I think the word kind of scares people. It's just going to give you some background knowledge on the idea. So the thing to remember here is you have to keep calm and create a hypothesis, right? This isn't something that you should really be stressed about. Another thing, going back to the middle school, and hopefully this is something that they did with you that you should remember, is the format for a hypothesis should be what we call an if-then format. If you're using the if-then format correctly, the if portion, this should refer to the independent variable, and the then portion will refer to the dependent variable. We'll spend some time going through and talking about the difference between these two things, independent and dependent variables, in a separate video, but the basic idea behind that is the independent variable doesn't change, the dependent variable does change. So for example, if I wanted to set up a very basic experiment, and I said that like all students will study for an hour in order to get ready for their quiz. So that would be the if part, right? If all students study for an hour to get ready for their quiz, then quiz scores will go up. So the if part is independent. That's the same for everybody. We'd say all of you guys have to study for an hour. The then part is the dependent part. That we're not 100% sure about. Maybe some people's quiz grades go up, maybe other people's don't. Maybe some people already had a high quiz grade, so there's really no way for their grade to go up. But the dependent variable is the one that's changing. Independent variable doesn't change. So everybody is studying for an hour. Dependent, though, is changing because you're going to have various results from that. The next step is to go through and gather data. Now, you'll be gathering some kind of data no matter what lab we're doing. Sometimes it falls into the traditional science gathering data picture that you probably have in your head. So here's this guy, he's looking at the results of some kind of test, and then this woman is recording it on a chart. Typically, you will have to record your data as we're going through and measuring things. For example, we'll do a lab later on in the year that has to do with fermentation, which is a process that your body goes through if you don't have enough oxygen, but you still need to provide energy for your muscles. And we'll actually go through it and watch yeast perform that process. But the way that works is that yeast produce carbon dioxide, amongst other things, as a byproduct, and we'll measure the effect of that carbon dioxide being produced because it'll be blowing up a balloon on top of a beaker that we're using in class. So that's something where you would be gathering a data point. Like every minute, you've got to measure the diameter of the balloon and see if it's increased. So that's the traditional kind of gathering data, getting the raw numbers from the experiment. In the next step, we'll actually be analyzing that data. So once we get to the analyzing stage, you're going to be going through and making sense of your data. So you'll probably have something like this, some kind of chart with your raw data in it. You then will either graph that or somehow represent that data in a way that makes it easier to interpret. It's generally not very easy to go through and interpret raw data in a chart, but once you graph it, it becomes a little bit easier to go through and see the trends and understand how the data is changing. So we'll go through and do this for every experiment that we're doing that has like graphable data. Some of them, again, I talked about like the DNA extraction. There's really not anything to graph there because we're not really dealing with a lot of numbers. You're dealing more with observations that you're making when you're going through and extracting the DNA. But if it's something that applies, I'll certainly walk you through how to do this and how to make graphs. We'll be using Google Sheets for all of this, and once you've done it a few times, it becomes very easy. The last step is your conclusion. This is the part that usually makes people nervous because you have to go through and write the conclusion at the end of the lab. I'll give you a clue now for the 3 steps that make writing a conclusion very easy. Because people always tell me, I struggle with this. This is generally the most difficult part. And I promise you, if you go through and use these 3 steps, this will become the easiest part of the lab. The first part is just how to get started. The thing that you do to start this one off is you go through and you restate the purpose of the lab. Why did we go through and do the experiment that you just completed? That's an easy way to get your conclusion started. During this lab, we were extracting DNA from cheek cells and from banana cells. Whatever it is that we were doing, you just restate that to begin the conclusion. The next thing is you're going to tell me what your hypothesis was. So you want to tell this one to me in if-then format. Here you're basically telling me what you thought would happen. This is your prediction. And then the last thing is your data. You want to tell me, well, what does your data say? Does your data support your hypothesis, or does your data support some other idea, some other conclusion at the end? One thing that's very, very important to remember, I'm going to write it up here in red. It's okay to be wrong. This goes back to your hypothesis. If your hypothesis is incorrect, that is not a big deal. It usually freaks people out because you guys are so used to everything having to be perfect and everything having to be right. It's okay if your hypothesis is wrong as long as you can tell me why. If you can explain why, then it is not a big deal at all. I'm actually more impressed with somebody with an incorrect hypothesis who can then go through and explain to me why it was wrong than somebody who did it right the first time. This one is very short. This does not apply to us, but something I want you to keep in mind is that the scientific method for scientists does not end at the conclusion step. Real scientists are going through and they're publishing their findings. This allows other scientists to learn from what they've done, and this is really how scientific knowledge builds. We'll go through a few examples of different experiments this year where actual scientists were reading things that other people had published, and that actually gave them ideas for how they developed their experiments. That's how we've gotten a lot of scientific breakthroughs, is from scientists reading the findings of other scientists. The last thing for us to talk about is a little memory trick. It's an acronym to help you remember the steps of the scientific method. So if we go through this one, for purpose and problem, we're going to think of Penargil. And we've got really has great air conditioning. So I know it's usually hot in the beginning of the school year, so this is one that most people find, somewhat memorable. But we've got the idea of the acronym here where each of these line up, and that should help you remember the different stages. So purpose and problem, Penargil, research, really, and so on. I do realize that this is bad grammar before you sick the English department on me, where we've got Penargil really has great air conditioning instead of has really great air conditioning, but this is the way that it works with our acronym. So if that's something that helps you remember those steps, it's a good little mental tool to have, just so that way you got the steps and that you have them in the right order. So as always, guys, I appreciate you taking the time to watch the video, and we'll talk about this one a little bit more in class.